Thank you. I'm going to start by trying to pretty much butcher a quote that should help you guys think about the way that we think about these kinds of problems. So the quote goes something like this. Greater fleas have smaller fleas on their backs to bite them, and smaller fleas have even smaller fleas, and so on ad infinitum. Okay? So what I want you to start to think about is the way that we look at the world ends up being through this process of understanding parasitism and infectious disease dynamics that are even deeper within some of those parasites. My title and what I'm going to talk to you today about is Eco-epidemiology, Endemism and Zoonoses. Those are large words that we should define really quickly. Eco-epidemiology really is using ecology in an epidemiological context so that we can take the tools of ecology, population biology, the understanding of dynamics within animals, and then use those tools to influence how it is that we either block the transmission of particular infectious diseases or in some cases just inform public health policy and folks like, for instance, at Flagstaff Medical Center in what is out there. Endemism and zoonoses. Endemism is this idea that these things exist here all the time. Okay, so that's something that is endemic. It's in your compost bin. It's in our backyards. It's at Grand Canyon National Park. And it's here widespread and really all over the place. And then zoonoses, which is really the kicker with this talk, is transmission of a pathogen from a wild animal to a human. Okay? And so something about and what has brought me to northern Arizona is the fact that we actually have a number of rare and dangerous pathogens in northern Arizona that are widespread and all over the place. So again, a manuscript was published a little bit ago in Nature, and what this manuscript wanted to do was map all of the emerging infectious pathogens that have been occurring over the last 50 or so years. This map shows you that something that you would probably think, a vast majority of these pathogens have shown up in circumtropical regions, right? Higher biodiversity with that fleas ad infinitum means that, guess what, we got way more bugs. At the same time, what they ended up seeing, that 70% of these emerging infectious disease events are the result of a zoonoses. So again, a pathogen coming from an animal to a human, and vector-borne. Vector-borne is the idea that an arthropod, or a flea, a tick, a louse, a mosquito, a biting fly, there's a whole long list of these things, transmit pathogens to people. Okay? If you further break up this data, looking on the x-axis, again, is the year, obviously, and then the number of EID events on the y-axis. And what you can see in panel A is that the majority of these things that we see are from bacteria, rickettsia. Rickettsia is really just another fancy, really small bacteria, and then viruses. There's also some worms and some fungus, but really the bacteria and the viruses are predominantly part of the EIDs. And then in panel B, what you see is that, again, just tabulating these EID events, they're coming from some zoonotic origin. Okay, so whether or not that's in the white directly from some wildlife species or in the others where it's either non-wildlife or unspecified. So non-wildlife is from our wonderful companion animals, our food, cattle, birds, what have you. Okay, so there's all sorts of times where we have intimate contact with these animals, and that intimate contact leads to infections. This is a little bit of a graphic of how we really like to see this. It's an additional reference from The Lancet, so one of the leading medical journals in the world, really. The Lancet, in this particular article, what they wanted to do was describe how it is that we as ecologists see this emerging phenomenon. And what you can see again along the y-axis is how we see this is in endemic pathogen cycles happen with their own dynamics. They're always cycling, they're very consistent, they never get really high, they never explode into what we would call a pandemic or a massive outbreak, but they kind of always exist at this particular level. Then, every once in a while, there's a spillover event. That spillover event is this pathogen that moves from some wild animal source into maybe our domestic animals and explodes into a massive outbreak, or, which can be even scarier, directly into people. And then what you end up seeing is this, this oscillation that's happening in wild animals, a spillover event that may go to a domestic animal that increases the amplification event, and in some cases spill over into humans, which then leads to, in red, what would be an outbreak. Okay. In our world, what we see this as is really that pathogens And these cycles exist completely independently of our existence. Whether we are here or not, they will still keep going. And we like to think about this because in our job, what we want to do is really be able to take that type of data, translate that into risk for humans or pets or critters that we actually care about. I always like to bring this up because the human factor really comes into play quite often. And that human factor is because we do put ourselves into harm's way, if you will. I took this picture a couple weeks ago. This is at the Grand Canyon National Park in the South Rim. Grand Canyon National Park has something like 2 to 5 million visitors a year. They are in this endemic region where we have a long list, ready? Plague, Rocky Mountain spotted fever, tick-borne relapsing fever, Hantavirus, what have you. There's a whole bunch of these things. And people still end up coming into contact no matter how many warnings we will give them, right? Some of us have probably done this, okay? This is what public health often sees, okay? So again, you can still see these endemic oscillations that are occurring along the bottom end of the graph. And what we in our field would use that is to be able to forecast an outbreak of some kind, right? Using these endemic oscillations to look at early detection and then really use that science-driven data to then inform public health policy and even folks in the clinic, okay? So again, this is what our lab does. Our lab goes out, we catch all these critters, we sample these critters where we're taking their ectoparasites off or we're sampling blood, and we're taking all of these other biological measurements, size, weight, what have you. We then extract the DNA, or RNA in the case of some viruses, and then we can run them on these fancy machines that really, they go bing, and then after they go bing, we can say whether or not that is positive, so infected with something, and then we can start to measure these dynamics. One of the bugs that my lab studies specifically is Borrelia hermsii. This is the causative agent of tick-borne relapsing fever. Its closest relative is Borrelia burgdorferi, which I'm sure many of you will know as the causative agent of Lyme disease. It's a spirochete, so it's a weird bacteria that really corkscrews through the tissue in Spirocatales. It is an obligate parasite, meaning that it can only live in a scenario where it is infecting something. Okay? It has a wide host range. It infects all sorts of wildlife species, and then ourselves, our pets, dogs, cats, etc. It's transmitted by Ornithodorus species ticks. These are a different kind of tick than you're used to thinking. The tick that you're used to thinking of feeds for maybe 6-7 days, gets really engorged, starts to look like a grape, falls off your animal, crawls under your couch, you never see it again, right? This guy feeds for maybe 20-30 minutes. Comes out of the woodwork or out of your bed or a nest, really, right, a nest. Comes out of there, feeds on you, you kind of think that you had a spider bite, and it's gone, okay? This is a map made by the Center for Disease Control and Prevention showing the high numbers of cases. This is actually why I live here. Okay, I can hear all the giggling. You're looking, oh, what's that one county? You're all from San Bernardino or something? No, Coconino, right? That San Bernardino map actually is mostly Big Bear, which is kind of interesting, and then the rest of the cases are, the majority of which are in the Sierra Nevada mountains. So Coconino, it's going on. So what we've been doing is we've established a series of these surveillance sites all over Flagstaff, including at the Arboretum, the experimental forest north of town, and then also in the Grand Canyon National Park, both the South Rim and the North Rim. So collaborations really with County Health, Arizona State Health, NAU, and then the National Park System in order to look again at this endemic dynamic to figure out where it is and why it's here. So this is about as much data as I'm going to show you, because most people look at the rodent community and they think, mouse, right? We see them slightly different. Okay, so when we trap these things, we're looking at them as this community of organisms, each that have different interplays with this infectious pathogen. This is our results, meaning that everywhere we look, there's a prevalence all over the place at about 3.6%, 3 to really 8% depending on where you look. And these two different species, the chipmunk and the wood rat, so the pack rat or whatever you want to call it, are the ones that are most infected with this particular pathogen. So we were doing all this work, and then lo and behold, there was an outbreak. Camp Colton, a lot of you probably know what Camp Colton is. There was an outbreak in a local football team where six high school students were infected with this bug. And because of the collaboration that we've had with Coconino County Health and with Flagstaff Medical Center, these guys were able to get treated really quickly and suffered from no disease. It doesn't mean that Camp Colton's a dirty place. Camp Colton's actually amazing. And so I don't know if you've been there, but it is an outdoor education facility that is an incredible resource for the community. What they were doing is getting rid of something even more dangerous. And so this next story that I'm going to tell you is really what leads to these nonlinear effects that we often see in ecology. So this guy. What they were doing is ridding the camp of Paramiscus maniculatus, or the deer mouse. The deer mouse is the main vector or the main reservoir host of Hantavirus, Endemism that was first discovered in the Four Corners. It is deadly. It has a case fatality rate of something like 40 to 52% or so, depending on what part of the world you check in. So it is incredibly important to get rid of. They got rid of the mouse. Now these cabins had nothing to feed on. The ticks still existed. They crawled literally out of the woodwork and fed, right, transmitting some pathogens. We do study Hantavirus cardiopulmonary syndrome in our lab as well. Again, we're looking at the dynamics within the deer mouse. Again, showing you our luck with infectious disease here in the Four Corners. Arizona happens to be one of the higher areas that you become infected with this pathogen. This is how you become infected with it. Really, it's the inhalation of fresh feces or urine in an enclosed space from a mouse that was living in that enclosed space. Okay, so just something to think about. We have done some extensive modeling with this pathogen where we looked at both its distribution on the landscape, looking at it spatially, and using those exact same surveillance sites from all over the place that I talked to you earlier. And we've been able to determine still that that pathogen also exists anywhere from 5 to 15% everywhere we've looked. Okay. So we made a couple of models. This is a model of the Four Corners. In red is where you would find the virus, and as it goes to green, the virus should kind of go away. It's a probability distribution. What the main take-home of this is that we mapped both the mouse and the virus, and what we see is that the virus, its distribution is nested within the distribution of the mouse. So the virus has its own biology, okay, its own biology, even though it's within predominantly the deer mouse. So science has a tendency to change policy, right? It also, especially when you deal with infectious disease, has a tendency to change your own actions. This is a representative field camp, if you will, of us pre-2012. This was in the Sierra Nevadas. Kind of good time. This is what we look like now. Right. This is a colleague, Marlene Gaither from Coconino County Health, and then my graduate student, Stephanie Sinkovich, and they're sampling at the south rim of the Grand Canyon. So even further, this is really what our setup looks like. It's kind of intense. There's a perimeter. You cannot go within, or you cannot move. If you are inside that perimeter, you can't leave it. If you're outside of it, you don't come into it. It's kind of a dangerous spot. You can see that we're wearing HEPA filters, so we have a filtered air that's coming through to us. This is our animal stock, so these are all the guys, little boogers that we caught that are in these little Sherman traps that we're pulling out, sampling, all catch and release. So we are marking them and letting them go. And then, importantly, this is bleach. Okay, this ends up spraying down everything that you see there. So again, I want you to remember these dynamics that we do and what what our work really entails really has to do with the oscillations within these wildlife populations. Okay? So we're looking at the dynamics, the biology of these wildlife populations, their then infection dynamics that is within those animals, and then trying to use that data to inform both public health policy, colleagues at Coconino County Health or Arizona Department of Health and the Center for Disease Control, as well as folks at Flagstaff Medical Center, so that they know that, which they do, know that these things exist here and they are widespread and relatively abundant. Really, who I would like to thank, again, are my colleagues from Coconino County Health and Arizona Department of Health, who without this work we would not have been able to get as much done and allow for the communication really between myself and and folks at NAU, and then the folks at Flagstaff Medical Center in order to inform, again, health policy. We get our funding from all sorts of different places, NSF, NIH, National Academy of Science, some private donors, and then the rest of these pictures are mostly of the grad students who really do actually all of the work. So, thank you.